Okay, so in this video, we're going to look at cells for AS for CA. And the first part of this really is looking at the different types of microscopes or microscopy. So the images that you see here, you can see an animal cell and a plant cell as seen under a light microscope. Okay, so light microscope's a very basic microscope, gives you less magnification and less resolution. Okay, so you can see an animal cell with a typical animal cell, the cell membrane around the outside, controlling what moves in and out of the cell. You've got your cytoplasm, where chemical reactions take place, and you've got the nucleus, which is surrounded by a nuclear membrane. Okay, and plant cells slightly different, slightly more complicated. Cell wall around the outside to give it a bit of protection, a bit of structure. Just inside the cell wall, again, you have the cell membrane. Again, plant cells have a cytoplasm where the chemical reactions take place. Within the cytoplasm, again, they have the nucleus surrounded with nuclear membrane. They also have chloroplasts, which absorb light for photosynthesis. And they also have a tonoplast or a vacuole, which is filled with cell sap. So that's how you would see an animal and a plant cell using a light microscope. Okay, now the units of measurement that we talk about when we use electron microscopes and in terms of microscopy in general, you have millimeters, okay, which are 10 to the minus 3 meters. So if you think of a meter as 10 to the 0, which would be 1, okay, millimeters are 1,000th of a meter, which is 10 to the minus 3. Micrometers are 1,000th of a millimeter, okay, which is 10 to the minus 6 meters. So a micrometer or micrometer with this funny U with an M beside it. And then nanometers, a nanometer is 1,000th, again, of a micrometer. So it's a 10 to the minus 9 meters, okay, nanometers, the smallest unit that we would use when looking at different samples. Okay, then we go on and talk about electron microscopes. Now, as it suggests, it's formed by shooting electrons through a very complicated microscope. We place our specimen on the grid up here. Electrons are shot out of, if you like, or they come from this cathode. They pass through anodes. The electron beam passes down through the sample, and then it forms an image, if you like, or the electrons meet at this fluorescent screen or photographic plate, and that can be viewed through the eyepiece. Okay, so it works by a beam of electrons, which have a shorter wavelength of light, and that gives you better resolution. So it gives you a clearer image, a sort of higher pixel image, and it also gives you a lot higher magnification. Okay, so the type of images you would see from an electron microscope would be here. You can see an image of pollen grains. So it gives you very, very detailed, high resolution images, much larger magnification, but the images are in black and white. Okay, so that's the main difference between, or one of the main differences between the light microscope and the electron microscope is black and white, much more resolution, much more magnification, more detailed image. Okay, then we go on and talk about cell ultrastructure. So we talk about eukaryotic cells in terms of animal cells, plant cells, and fungi, and then we'll talk a bit about prokaryotic cells towards the end. So if we start off looking at the ultrastructure of animal cells, okay, you can see here a typical animal cell. So as we talked about, that cell surface membrane, a bilayer around the outside. You've got your cytoplasm, which contains all the other organelles and where the chemical reactions take place. We'll look at these in detail in a moment. You've got the nucleus here with the nuclear membrane around the outside. You've got different parts of the nucleus, the nucleolus, the heterochromatin, the euchromatin, as we'll look at in a bit of detail at the moment. Okay, in a moment. You've also got mitochondria, which you can see here, this sort of shape. Lots of mitochondria, that's a cross section, so cut at sort of different angle. You've got Golgi apparatus. You've got the rough endoplasmic reticulum. You can see with that rough edges to it. You've got the smooth endoplasmic reticulum. You might have some glycogen granules, some sort of stored glucose there as well. You can see some centrioles here as well, some little vesicles. You might have lysosomes that we can see here, some free ribosomes. All of these organelles you need to know in detail. Okay, so the first one we'll take a look at is the nucleus. Okay, so the nucleus has that membrane around the outside, that sort of bilayer or nuclear envelope. In between, it has small gaps called the nuclear pores, which allow messenger RNA to move out and other things to move out as well. The more dense parts of the nucleus are called heterochromatin, and the less dense parts are called euchromatin, as you can see there. Okay, you also have the nucleolus, and the job of the nucleolus is to produce ribosomes. Okay, and the ribosomes then form on the rough endoplasmic reticulum, which you can see just forms outside the nuclear membrane, as we'll talk about in a second. Okay, so the nucleus or the DNA is wound around proteins called histones, and that is a bit like a fishing reel, and the DNA gets wound around it very tightly, and it allows it to store a lot of DNA within a very small space. Okay, so nucleus tends to be between 10 and 25 micrometers, so 10 to the minus 6 meters in diameter. Has that nuclear envelope or double membrane around the outside, which also has the pores. It contains DNA in the form of chromosomes, which are wrapped around these histones. Okay, and when not dividing and the chromosomes aren't visible, it forms chromatin. Okay, and you have different types, as we talked about there. The densely darker bits are called heterochromatin, and the less dense bits called the euchromatin. Okay, the outer membrane is covered with ribosomes, which is where the rough ER originates from. And inside the nucleolus, as we talked about, you have, or inside the nucleus, you have that nucleolus, and that's where the ribosomes are formed. Okay. Another diagram just showing the nucleus there. You can see the nucleolus, that double nuclear envelope around the outside with the nuclear pores, that heterochromatin, the dark forms, euchromatin, the lighter parts of the nucleus. Okay. The next part then is looking at the rough endoplasmic reticulum, or the RER. Okay, so that was just outside the nucleus that you can see here, almost forming from those pores, or the double membrane, and it sits outside, and it has lots of these ribosomes attached to it. Okay, and the ribosomes are really to do with making proteins, so protein synthesis. So that's the main job of the rough ER, is to make those proteins. Okay, they're transported or distributed throughout the cell. It's continuous with the nuclear membrane. It also makes it easier to transport messenger RNA, which we talk about more at A2 in terms of protein synthesis. And it's found in cells that make lots of proteins, so things like pancreatic cells, which make lots of digestive enzymes. Okay, the smooth endoplasmic reticulum, or SER, is a system of tubes, membrane tubes in the cytoplasm, and that is really where lipids, fats, and steroids are made or metabolized, and also to do with cholesterol metabolism as well. Ribosomes, you can see here, tend to have a small diameter, about 30 nanometers, so that's 30 times 10 to the minus 9 meters. They're non-membrane bound organelles, so no membrane around them. They are attached to the rough ER or can be found free in the cytoplasm, and they're made up of a small and large subunit, and they are the site of protein synthesis. So those are the sort of protein makers within the cell. So they would make hormones, make structural proteins, make enzymes. Okay, then you have the Golgi apparatus, which is that curved stack of flattened membrane. Okay, and it forms small vesicles containing protein, which pinch off the rough ER, and then they join at the cis or convex surface of the Golgi apparatus, which is closest to the nucleus. The Golgi apparatus is really involved with the modification of proteins. So it will convert them into glycoproteins or specialized enzymes, etc. And then vesicles, which contain that modified protein, but off the concave or the trans surface, which is further away from the nucleus. And then that vesicle transports proteins to different parts of either the cell, or they may be used with other cells or move out of the cell through exocytosis. Okay, so there, if this was up to the side of the nucleus, you have vesicles containing protein coming from the rough ER, which join on the cis surface, concave surface. Those are then modified within the Golgi apparatus, and then the modified proteins form of the in vesicles of the trans face, so that convex surface further away from the nucleus. And as I say, they can move outside the cell or be used inside the cell as well. Okay, so there you can see the rough ER making the proteins, join on to the cis surface, or that concave surface, and then when they're modified, they form vesicles, those modified proteins, and they may be got rid of to move to different parts of the cell. Okay, and you can see that in this little animation. So here we can see the rough ER up top. The proteins are made within the rough ER. They're little vesicles bud off the rough ER and move towards the Golgi apparatus. Okay, and that forms at that cis end. And then those proteins will be modified within the Golgi apparatus. And then at the trans end, further away from the nucleus, those modified proteins again will bud off with a little vesicle forming around, or a little double membrane forming around them. And it may be used within the cell, or may be got rid of by exocytosis, as you can see there. Okay, lysosomes are the next one that we'll take a look at. So lysosomes are about 0.5 micrometers, or 0.5 times 10 to the minus 6. They're formed by the Golgi apparatus. They contain hydrolytic enzymes for use within the cell, so they digest either worn out organelles, or may be involved with breaking down and engulfing bacteria in phagocytes or phagosomes. They have a thick membrane which prevents the enzymes being accidentally released within the cell, or which may cause damage within the cell. And we can see how lysosomes work here in terms of the vesicles bud off that Golgi apparatus. You can see there it forms these enzymes or lysosomes with the hydrolytic enzymes inside them. They may be used to digest remains of parts of cells, or broken down organelles, or foreign bodies in terms of bacteria, etc. as well. And if we watch this little animation, this describes as well what they do.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. These enzymes digest particles or cells taken into the cell by phagocytosis. They also digest old organelles such as mitochondria. The hydrolytic enzymes that degrade proteins, nucleic acids, lipids, and carbohydrates are formed in the endoplasmic reticulum and then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles such as defective or worn-out mitochondria. This results in the destruction and recycling of these structures.
Okay, so that's how lysosomes work. As it mentioned there, mitochondria, that's the next one we look at. So they're about 10 micrometers, or micrometers in length. So that's 10 to the minus 6 meters. Again, they have a double membrane, as we'll see in a moment. You have the outer membrane and inner membrane, and in between that, you've got the intermembrane space. The fluid-filled matrix is the sort of cytoplasm, if you like, of the mitochondria. And then you've got the inner membrane is highly folded into things that we call cristae. Okay, and that gives the mitochondria, the internal part of the mitochondria, that large surface area. Okay, they also have enzymes on the cristae which we know is ATP synthase, which are involved with aerobic respiration. Okay, so they produce ATP, which can be used for different types of energy requiring reactions within the cell or around the cell. So they're very metabolically active. Okay, cells are metabolically active, sorry. They will contain many mitochondria. Okay, so that ATP can be used with active transport. It gets used with nerve impulses, used with muscle contraction, etc. Okay, so those types of cells will have lots of mitochondria. Okay, so there's the mitochondria. You can see the outer surface. The inner surface is highly folded with these cristae. You've got the intermembrane space here is this sort of sandy colored, and then the matrix is that bit inside the inner membrane, the sort of cytoplasm of the mitochondria. Okay, the next one then that we look at are the microtubules. Okay, so these are hollow cylinders made of protein, a particular type of protein called tubulin. They tend to be about 25 nanometers in diameter, so very, very small, 10 to the minus 9, 25, 10 to the minus 9 meters, and can be up to 10 micrometers. That's 10 to the minus 6 meters in length. Okay, they occur as a pair of centrioles in the centrosome, which is just outside the nucleus. And then those centrioles are a pair of short microtubules, which are positioned at right angles to each other. Okay, and it's these centrioles that will move to the opposite poles of the cell and produce the spindle fibers, which are involved during cell division. Okay, also found in cilia and flagella, which allow certain cells to move around, and also as part of the cytoskeleton. Okay, they're not found in plant cells, as you can see there. Okay, then if we look at plant cells, you can see, as we talked about, that cell wall around the outside. They have the nucleus, nucleolus, and they have the rough ER. They have the Golgi apparatus, so protein synthesis, modification of proteins. They have that ATP producing mitochondria, many of those. They have those smooth endoplasmic reticulum for those different types of things involved with that. So in terms of producing lipids and phospholipids, etc. And then also we have these chloroplasts, which we'll look at in detail as part of this as well. Okay, between plant cells as well, you have slight gaps in the cell wall called plasmodesmata. Okay, and where plant cells join together, you have that sort of cement that holds them together, which is called the middle lamella, that we'll talk about in a bit of detail as well. Okay, cell wall is mainly composed of cellulose. It tends to be about 1 micrometer thick. It's a very rigid structure, very strong cellulose, beta glucose molecules, hydrogen bonds give them that structure and strength. It's very important in terms of turgor, or when the cell fills up with water, it stops it from bursting. And also it prevents that protoplast, restricts the outward expansion of the protoplast or the cell membrane as the cell takes in water. Okay, it's fully permeable, so all substances can pass through the cell wall, and it's the cell membrane which gives it that selective permeability of the plant cell selective permeability. There you can see the cell walls around the outside. They have these plasmodesmata, which allow water and different molecules to pass between cells, and you have that middle lamella down, the sort of cement which holds the cells together as well. Okay, the plasmodesmata are those strands of cytoplasm that extend between cell wall of neighboring cells. The plasma membrane is one plant cell continuous with the next, and it allows substances to pass easily, as we talked about there. So you can see there, you have that plasmodesmata allowing substances to pass from cytoplasm to cytoplasm, from cell to cell. Okay, the chloroplast then is really involved with photosynthesis. Okay, again, similar to the mitochondria, it has an outer membrane and an inner membrane. Okay, so it's got that double membrane or double envelope. Inside, you've got what's called the stroma, and that's a bit like the cytoplasm of the chloroplasts. Within that, you've got a flattened, or a network, as it talks about, a flattened membranes called thylakoids. Okay, and those are often referred to as sort of pennies or 2p's stacked on top of each other, and each of those stacks form something called a grana. Okay, and between the grana and the other membranes, you have intergrana, which we'll see in the diagram in a moment. And it's those thylakoid membranes which contain the chlorophyll, which absorbs light for photosynthesis. Okay, you may also have starch grains, lipid droplets, which are products of photosynthesis as well. And as it says, excuse me, the chloroplast is the site of photosynthesis. So there's your double membrane around the outside, intermembrane space in between. You've got your starch grains, lipid droplets, which are products of photosynthesis. There you can see you've got these thylakoids, these flattened membranes, sort of like your 2p's, your pennies stacked on top of each other. There's your intergranal lamella, which join the grana, which is these stacks and the grana together. Okay, and then that sort of cytoplasm, as we call it there, is known as the stroma. Okay, the next bit then really is looking at fungal cells, okay, or fungi. They are similar, but obviously slightly different as well. They have a vacuole in the center, and you can see they may have a couple of nuclei or 2 nucleuses, if you like. And they have mitochondria as well. They may have glycogen granules, so those stored for stored glucose. They have rough endoplasmic reticulum, so they obviously produce proteins, and they have the smooth endoplasmic reticulum for making lipids and cholesterol and things like that as well. Okay, cell wall around the outside, you can see the cell wall is non-cellulose, so it's made of a substance called chitin, which again is a strong glycoprotein which holds the cell together, and they do have lysosomes and things as well involved with them too. Okay, so they are plant-like structures. Okay, they have cell walls made of chitin. They do possess vacuoles. They do have animal-like structures in terms of glycogen and lysosomes. The cells form threads called hyphae, and they may be, as we talked about there, multinucleate. Okay, so they may have many nuclei. Okay, there is a little chart or a little table. Sometimes you get these types of things in exam questions. You have to fill in your ticks and crosses, etc. So animal cells don't have a cell wall. Plant cells, cellulose cell wall. Fungal cells have a cell wall made of chitin. Plant cells are the only types of cells which contain chloroplasts. Centrioles are only found in animal cells. Lysosomes, so those digestive enzymes within the vesicles, are only found in animal and fungal cells, not in plant cells. Animals store carbohydrate as glycogen, as do fungi. Plants will store it as starch. Okay, so that's the sort of stored glucose in case the cells need it. And then all 3 types of cells have a nucleus. Fungi are slightly different in that they may be multinucleate. Okay, so those are all the eukaryotic cells. Prokaryotic cells are slightly different. They're seen as sort of simpler. These would be older cells, if you like, in terms of their ancestry and where they've come from. They're more basic type cells. Okay, so they do have a cell wall, which is made of a glycoprotein, which can be surrounded sometimes by a slime capsule around the outside. Okay, they do have ribosomes, so they do make their own proteins. They tend to be smaller ribosomes than what you would find in eukaryotic cells. They have storage molecules in terms of glycogen or lipid stores. They do have a cytoplasm. They have circular loops or circular bits of DNA called plasmids, which are useful when it comes to genetic engineering that we look at in A2. And they have free DNA, or what we call naked DNA, which isn't surrounded by a nuclear membrane. Okay, so it's just free DNA which you find within the cell. Okay, there you can see the sort of difference between prokaryotic and eukaryotic cells. So prokaryotic cells tend to be a lot smaller, no nuclear membrane, and they have small looped bits of DNA known as plasmids. The ribosomes are smaller. They don't have those complex organelles in terms of the mitochondria or Golgi that we find in eukaryotic cells. A reasonably complex cell wall made of a glycoprotein that you can see there. The ones we've talked about there, you can see with the eukaryotic cells. Plasmids usually present, not present in eukaryotic cells. Microtubules not present in prokaryotic cells, but are present in some of the eukaryotic cells. Okay, then we go on and talk about the cell membrane in terms of that phospholipid bilayer. So if you remember, the phospholipids are made up of two fatty acid tails, your glycerol molecule, and a phosphate, which has replaced one of those tails from the triglyceride. So that forms a phospholipid. The head of the phospholipid is known as hydrophilic. Okay, so that sort of means water liking, if you like. And the tail is hydrophobic, so it doesn't mix with water or doesn't like water, if you like. Okay, more detailed structure you can see here. So you've got that phospholipid bilayer. Okay, so the heads on the outside, tails found on the inside. You may have transmembrane proteins or integral proteins within the phospholipid bilayer. You have cholesterol within the bilayer, which is involved with the fluidity and involved with the structure of it. The glycocalyx is the outside of the membrane, so this would be the inside of the cell. These are found on the outside, and some of these are recognition sites. Some of these allow different molecules to pass in. So you can see here you might have glycoproteins. Okay, so a protein combined with some sugar molecules. You have some glycolipids. So there's your sugar molecule joined to a phospholipid. You have some extrinsic proteins possibly as well. And these transmembrane proteins tend to allow substances to pass through. Okay, and that gives you a bit of detail there, a bit more detail about the plasma membrane. So pause and have a look at that if you want to. Then we go on and talk. The last bit of this is really looking at the structure of viruses. Okay, so viruses are very different in terms of their structure. They're not living cells as such, in that they need a host to be able to survive. And the way they get into the host, you can see here demonstrating on the left. So they have a DNA molecule, again, naked DNA found within a protein coat or a protein head that you can see here. They have, excuse me, a contractile sheath, which is made of protein. The tail plate, or the tail fibers on a base plate. This is an example of bacteriophage. So this is a virus which infects bacteria and causes bacterial cells to be destroyed. And they sort of inject their DNA, if you like, into the bacteria. We'll go back and look at that again. So you can see here that the virus will inject its DNA into the bacterium. It takes over the bacteria and produces new viruses and then destroys that bacteria. Okay, HIV is another virus that you need to be aware of. So HIV is something that we call a retrovirus. Okay, so rather than having DNA inside it, it has RNA, and it also has reverse transcriptase. And the reverse transcriptase converts that RNA into DNA when it invades a cell. And then that DNA takes over the cell and produces new HIV viruses. Okay, you can see it's got a slightly different protein coat around the outside in terms of glycoprotein. So it's bits of sugar here as well. And then it's also got proteins within the inside. Okay, and the last bit of this is looking at how it reproduces. And we'll take a look at this little animation.
AIDS is caused by the human immunodeficiency virus, HIV. This is an enveloped virus. The envelope is derived from the host cell membrane with viral glycoproteins such as GP120 and GP41 inserted into the membrane as the virus leaves the cell. The core is composed of P24 capsid protein, and the P17 matrix protein helps maintain viral structure.
Don't need to know that detail.
Inside the core are 2 identical copies of single-stranded RNA viral genome and 3 enzymes: reverse transcriptase, RT, protease, PR, and integrase, IN. To establish infection, HIV must first attach to its host cell. Attachment occurs by interaction between GP120 on the surface of the virus and the CD4 antigen receptor on the surface of the host cell.
This will be one of these little recognition sites that we talked about in terms of glycocalyx. So it may be proteins or glycoprotein on the outside. It tricks its way, if you like, into the cell using these recognition sites.
In addition to the CD4 receptor, there must also be a co-receptor on the host cell. The co-receptor differs for different host cell types. In T lymphocytes, the co-receptor is called CXCR4, whereas on macrophages, the co-receptor is called CCR5. After attachment, the viral envelope and host cell membrane fuse, resulting in entry of the virus into the cell. Once the RNA is released into the cytoplasm of the host cell, reverse transcriptase makes a DNA copy of the viral RNA genome. As the DNA is being formed, reverse transcriptase degrades the RNA strand. A complementary DNA strand is then added by the reverse transcriptase, and the ends of the resulting double-stranded DNA segment are joined non-covalently. Treatment with nucleoside analogs or reverse transcriptase inhibitors interferes with these steps. The resulting circular DNA is then moved to the nucleus and inserted into the host cell chromosome by the viral integrase IN enzyme. The integrated viral DNA is now referred to as proviral DNA. Following integration, the proviral DNA may remain dormant, or with host cell activation, RNA may be synthesized from the DNA, yielding messenger RNA and viral genome RNA. Viral messenger RNA is translated, yielding viral enzymes and structural proteins. Some of the functional proteins are formed by cleavage of a long polyprotein by the enzyme protease. Protease inhibitors interfere with this step. GP41 and GP120 are inserted into the host cell membrane, and the structural proteins surround the viral RNA to form the core. Finally, the virion is released by budding.
Okay, so HIV viruses in humans, they affect white blood cells, special white blood cells called lymphocytes, which are involved with your immune system, or helper T cells, specialized lymphocytes. So therefore, HIV attacks the immune system of humans. Okay, unfortunately, no cure at the moment. Retroviruses are very hard to find cures for. Okay, hopefully that helped with AS cells.